Bitter foods and drinks are generally not liked by humans, however coffee is still one of the most popular drinks in the world. There is a lot that goes into brewing a perfect cup of coffee. Mostly this has to do with roasting degree and caffeine content. And how people take their coffee varies greatly. Some like it milky, some like it very sweet, and some like it black, rich and strong. So what are the things that influence how much coffee we drink and how we drink it? Number 1. Behavioural effect of coffee. Caffeine is a stimulant of the central nervous system and metabolism. It promotes wakefulness, enhances mood and cognition, and creates stimulatory effects. These effects are linked to the development of flavour preferences. Number 2. Metabolism rate of caffeine. Caffeine is converted mostly to paroxetine by cytochrome P450 enzymes. These enzymes are coded by the CYP1A2 gene. If you have the C allele of this gene, you will be a slow metabolizer. If you have the A allele of this gene, then you will be a fast metabolizer. If slow and fast metabolizers drink the same amount of coffee, slow metabolizers will be exposed to higher caffeine levels. So if you are a fast metabolizer, you are likely to drink more coffee. Number 3. Bitterness responsiveness. Your preference for coffee is also linked to genetic variations in taste responsiveness. Each person responds differently to astringent stimuli. This influences our perception of bitter taste and our use of sweetness in our coffee. Low responsiveness to bitter taste means you will be more likely to drink coffee. So the aim of this particular study is to investigate the possible relationship between a person's caffeine metabolism rate and their bitterness responsiveness, and to see how these factors influence how much coffee a person drinks and their preference for coffee in terms of sensory properties and their need for sweetness.
Methods. 135 people were paid for their participation in the study. Of these, 59 were male and 76 were female. Of the females, 19 used oral contraceptives, and of the total number, 27 were smokers. There was no mention of the recruitment method. 6 samples of coffee were selected for to include a range of different characteristics. Importantly, aroma was tested at 65 to 67 degrees compared to 55 to 57 degrees used for tasting. This is because the higher temperature can scald the tongue and limit the taste perception of the participant. 6 equi-intense samples of caffeine and quinine were compared for their bitterness intensity. A single solution of pork was also used. Those who can taste pork typically have the bitter taste receptor TAS2R38, which is associated with a greater sensitivity for bitterness. 4 separate evaluations were undertaken during the study. For these, the general labelled magnitude scale was commonly used. It is an anchored line scale with quasi-logarithmic spacing and marked with verbal descriptors of intensity, such as strong and weak. CYP1A2 activity was determined by either genotyping or phenotyping. 37% of participants agreed to DNA testing, and the genotype of the SNP RS762551 was measured, as it is associated with speed of caffeine metabolism. Phenotype was measured by enzymatic immunoassay of saliva samples. Prior to testing, it was suggested that participants avoid certain foods, liquids, and activities, as these could inhibit or upregulate CYP1A2 activity. A pilot test was initially undertaken using 10 participants. The absorbance of saliva samples was measured at baseline and several times following a caffeine intake. As no variations in caffeine content occurred between 90 and 270 minutes, with a wider scale test, measurements were only done up to 90 minutes. was expressed as a caffeine metabolism index, which is a ratio of the residual caffeine following metabolism and the amount at baseline. The higher the CML value, the faster the caffeine is metabolized by the participant in the hour following the absorption peak. Flavour preference was measured by asking participants to choose between two commercially available packets of coffee, advertising either a strong flavour or a more moderate flavour. The consumption questionnaire asked participants how often they consumed coffee and in what common form. Perceived effect was also measured on a 7-point scale, beginning with strongly agree and finishing with strongly disagree. Examples of statements included, "Coffee improved my concentration" and "Coffee makes me jittery." A variety of data analysis methods were used, including unpaired t-tests, both 1- and 2-way ANOVA, and chi-squared tests.
Firstly, a high caffeine metabolism index group and a low caffeine metabolism index group needed to be defined, and this was done based on Z-scores to take into account that there was a significant effect of sex on caffeine metabolism index. The low caffeine metabolism index group had a Z-score less than the median of 0.0056, while the high caffeine metabolism index group had a Z-score above 0.0056. The median Z-score was excluded from analysis. It has been found in previous literature that oral contraceptive users and smokers have altered caffeine metabolism rates. Therefore, these groups were excluded from analysis. It is important that there is no significant differences between the two groups at baseline to rule out possible confounding effects. There was no significant differences between groups in sex, age, prop intensity, which is a bitter compound with no psychoactive effect, BMI, or fungiform papillae numbers. As can be seen on this graph, the low caffeine metabolism group perceived coffee as more bitter compared to the high caffeine metabolism index group. This result was significant at the 5% significance level. There was no difference in bitterness intensity of prop or quinine hydrochloric acid between high and low CMI groups. These compounds do not have a psychoactive effect, therefore the bitterness effect is exclusive to caffeine. This is evidence that the bitterness effect is mediated by caffeine's psychoactive effect. No difference in coffee liking was found between slow and fast caffeine metabolizers. However, slow caffeine metabolizers added on average 0.8 grams more sugar into their coffee, which was statistically significant. Also, more participants in the high CMI group consumed coffee regularly without sugar compared to the low CMI group. As can be seen on this graph, a significant positive correlation between caffeine metabolism index and daily coffee consumption was found, with an R-squared value of 0.9344. Based on previous literature, we would expect the AA genotype to have a high CMI score and the CC genotype to have a low CMI score. However, as can be seen visually, this is not the case. Upon one-way ANOVA analysis, a p-value of 0.569 was found, which indicates a non-significant relationship. Firstly, the small sample size and not everyone agreeing to genetic genotyping would have likely affected these results. Additionally, environmental and endogenous factors are likely at play. And thirdly, no single nucleotide polymorphism in the gene has been specifically identified to predict metabolic phenotype. Further research could stem from this study. Firstly, the study should be repeated to ensure reproducibility. Additionally, the biology of the bitterness response is unclear, and it is also unknown the exact cause of differences in caffeine metabolism rate.
In doing this experiment, the researchers were aiming to explore the relationships between caffeine metabolism, coffee flavour preference, and coffee consumption. The experimenters hypothesised that individuals who metabolise caffeine quickly and could tolerate the bitter flavour of coffee are more likely to consume more coffee on a daily basis. This was primarily tested using enzymatic immunoassay of saliva samples collected post-caffeine ingestion to determine a caffeine metabolism phenotype, defined as either high or low caffeine metabolism index. This phenotype is dependent on the expression of the CYP1A2 gene. This data was supplemented with flavour and aroma evaluations, along with questionnaires determining coffee consumption habits and flavour preferences. One of the main highlights of this study was the connection made between the rate of caffeine metabolism and the perception of bitterness in coffee and caffeine solutions. From this connection, the experimenters deduced that the individuals with a low caffeine metabolism index score were more opposed to the bitter flavour of the coffee and caffeine solutions. This explains why the slow caffeine metabolizers chose to add more sugar to their coffee than the fast caffeine metabolizers. The experimenters noted that the addition of sugar was used to mask the bitter taste of the coffee. It was also concluded that daily coffee consumption habits were less regular in the participants with a low caffeine metabolism index. The only significant difference in bitterness responsiveness between high and low caffeine metabolizers was observed after ingesting the samples containing the stimulatory compound caffeine. This suggests that the psychoactive effect is what determines differing levels of bitterness perception in individuals. These findings propose key ideas such as post-ingestive effects, flavour preferences, and caffeine metabolism for further investigation in future studies. The current study used an entirely Italian cohort to obtain roughly 90% of their daily caffeine intake from coffee. An option would be to repeat the study with a new cohort who drink less coffee and see if the results are still the same. Another possibility is to repeat the investigation using another caffeinated beverage, such as energy drinks, and see if this yields similar results. Finally, when repeating the study with energy drinks, it would be interesting to see if the presence of sugar affects preference and consumption habits.